My dear students, welcome to all of you. Today I am going to deliver a lecture on Idogonium, its important features and life cycle. First, we will take the classification of Idogonium. Idogonium belongs to the class Chlorophyceae, order Odogoneales, family Odogonaceae. The family comprises about 400 species and 3 genera. The 3 genera including in this species are Odocladium, Bulbocate and Odogonium. The Odogonium only possesses 285 species, that is why it is the type of the family. Now we will take the habitat of the genus Idogonium. The habitat of the Odogonium is freshwater. It usually prefers quiet conditions, quiet waters and avoid flowing waters. It is attached with the substratum by means of a rhizoidal cell known as holdfast. It also grows epiphytically on larger green algae leaves, petioles, stamens of the aquatic angiosperms. It is also found in some quiet situations of ponds, lakes and streams. The thallus structure of Idogonium is filamentous thread-like. The cells in the filament are placed end to end and the rhizoidal cell is fixed with the substratum. The free end of the apical cell is somewhat rounded and extended. The cell structure of Idogonium is like this. The cell wall is conspicuously or inconspicuously made of 3 layers. The innermost is cellulose layer, middle pectic layer and outer chitinous layer. The outer chitinous layer prevents the loss of the pectic layer, so the filament looks like a wet thread. Just inside the cell wall there is cell membrane and the protoplasm is constituted of a large central vacuole which possesses the cell sap. The protoplasm also contains a large reticulate chloroplast in which are embedded a large number of pyrenoids. A parietal nucleus is also present in the cell. Certain cells in the filament possess ring-like markings of cellulose which are 1 or 2 in number. These cellulose markings are known as caps and the cell containing these caps are known as cap cells. Now we will discuss about the growth and development of Idogonium thallus. The growth usually occurs simply by cell division. Usually in Idogonium, certain cells in the filament which possess caps or cap cells, they undergo cell division. Not all the cells in the filament undergo cell division. It is only that those cells which possess the caps or called cap cells which undergo cell division. The cell division occurs here in 3 steps. First is ring formation, next is nuclear division and third is formation of new cells. The ring formation occurs simply by the formation of a scar-like structure just at the upper end of the cell. This scar gradually thickens and forms a ring-like structure. This is of a hemicellulose in nature. The nuclear division is this ring-like formation is followed by the nuclear division. The nuclear division starts by the movement of nucleus towards the upper end of the cell where it divides into two daughter nuclei. The nuclear division is followed by the formation of cytoplasmic strand between the two nuclei. The formation of two cells occurs by the breakdown of the outer two layers outside the groove, just outside the ring. And this breaking of the two outer layers permits the ring to stretch out. Now the cell is twice as long as the normal cell, and finally the cytoplasmic strand moves upwards just at the base of the groove, and finally the cell gets divided. In case of Oedogonium, the successive divisions takes place only in the cap cells, so the number of caps formed on the cap cells determines the number of cell divisions cap cell has undergone. Now we will discuss about the reproduction of Oedogonium. Oedogonium reproduces usually asexually by fragmentation and spirulation, and sexually by advanced oogamy. The fragmentation in case of Oedogonium takes place either by dying off some of the cells of the filament or by accidental breaking. The fragments so formed by growth and cell division develop in the new plant bodies. In asexual reproduction, the zoospore formation is most common form, where certain cells of the filament, usually the cap cells, they develop into zoosporangium. Each zoosporangium produces only one zoospore. At the formation of zoospore, usually the cytoplasm recedes from the cell wall and gets collected in the middle of the cell. One portion of this zoosporangium, one portion of the cytoplasm present in the zoospore recedes and becomes hyaline, colorless. At the base of this colorless beak, a ring of flagella is formed. This ring of flagella are produced from the blephroplasts. So these blephroplasts are arranged in a ring-like structure. These blephroplasts are connected by a fibrous filament, so forming a circular ring. So from this blephroplast ring arises a single flagella from each blephroplast. So a ring of flagella is formed around the zoospore at the upper end, just below the beak. These zoospores are liberated out from the zoosporangium by the transverse rupture of the zoosporangium. On liberation, they move into the water for some time. After their movement, they settle down on the solid substances where they germinate. Before germination, they attach themselves with the substratum by this colorless beak, and this colorless beak gets converted in the rhizoidal cell, that is also known as holdfast. The remaining part of the zoosporangium divides transversely and develops into the new filament of the thallus. This zoospore formation usually takes place during the favorable environmental conditions. Now we will take the achinids. The next type of spore which is formed in the Oedogonium filament during unfavorable conditions are called achinids. These achinids are thick-walled structures and are produced singly in some of the specialized cap cells of the filament. These achinids on maturity also are released in the same manner as the zoospores are released from the filament and develop into the new organisms on the approach of new favorable conditions. Dear students, now we will discuss about the sexual reproduction. In Oedogonium, it is distinctly advanced type of oogamy. The sexual reproduction is, in case of Oedogonium, is by the formation of special reproductive organs, that is antheridia and oogonia. these Antheridia and Oogonia are developed by the special cells of the filament. So not all the filaments, not all the cells of the filament produce Antheridia and Oogonia, but certain specialized cells having usually one or more cap cells at their apical ends which develop into the gametangia. Sexual reproduction in case of Oedogonium is determined by certain external factors such as sunlight, concentration of carbon dioxide, and the deficiency of nitrogen. Depending upon the distribution of sex organs, the Oedogonium has been divided into two species, Macandrous species and non-androus species. The two species of Oedogonium can be differentiated from each other by the possession of small dwarf male filaments in case of non-androus species, whereas these dwarf male filaments are not found in Macandrous species. In Macandrous species, usually the Antheridia are produced on the filament of the normal size, whereas in case of non-androus species, the Antheridia are produced on small dwarf male filaments. The Macandrous species may be monoecious or dioecious. In case of monoecious species, both Antheridia and Oogonia are developed on the same filament. That means the filaments are bisexual. Whereas in case of dioecious species, the male and female gametangia or Antheridia and Oogonia are developed on the separate filaments. Whereas in case of non-andria, the Antheridia are produced on the small filaments called non-andria or male filaments, which remain epiphytically attached with the Oogonium or suffultatory cells, which is present just below the Oogonium. Thus, in non-androus species, there is dimorphism in case of Oedogonium thalli. That is, male and female Oedogonium filaments are different in shape, and they are different also in their physiology. Now, dear students, we will discuss the development of Antheridia and Oogonia in case of Oedogonium. The Antheridia or the male reproductive organ is produced by repeated cell division of any of the cap cells of the filament. Usually the cap cells divide vegetatively or divide by usual mitotic division into so many cells transversely. Each segment or each cell divides transversely or vertically into two sperms. The sperms after their formation are just like zoospores in their morphology, but they are smaller in size and having similarly the crown of flagella at the base of the beak-like structure. The sperms are liberated simply by the transverse division or by the transverse rupture of the antheridium, similarly as the zoospores are liberated. Now these sperms swim freely in the water for about an hour. Oogonia are highly differentiated female gametangia. The Oogonia develops from the actively dividing or actively growing cap cell, and this growing cap cell acts as oogonial mother cell. This oogonial mother cell divides vegetatively or mitotically into two cells. The upper daughter cell is having more cytoplasm and larger nucleus, acts as oogonium, and the lower cell acts as suffultatory cell or supporting cell. The cytoplasm of the oogonium stores reserved food material and a large amount of cytoplasm and develops into oospore or egg. In the Macandrous species, the male and female gametangia are developing on the same filament. To ensure cross-fertilization, the male gametes or antheridia are getting matured just one day after the oogonia to ensure cross-fertilization. Whereas In Meiosis species, the male and female gametangia are born on two different filaments. Now, sexual reproduction in Nenandra species. In Nenandra species, the sexual reproduction is somewhat specific and complicated. Here, the male gametes or antheridia are produced on small dwarf filaments called Nenandria. These Nenandria are actually produced by the special type of spore called androspores. These androspores are produced by any of the vegetative cells of the oedogonium, but the vegetative cell which develops the endospores usually possesses one or more caps at their anterior end. These cap cells divide transversely into so many cells, and each cell develops a single androspore. The androspores are identical like zoospores, having same crown of flagella at the base of the beak, and they are produced, liberated in the same manner as the zoospores are liberated. When these androspores are liberated by the rupture of the androsporangium, they move freely about in water medium, and finally they reach the female filament of the oedogonium thallus. In Nenandra species, after attaching themselves with the egg or oogonium or supplementary cell of the oedogonium, they germinate. After germination, they attach themselves with the oogonium by the rhizoidal cell or the holdfast, and the remaining portion gets divided by transverse division into so many cells. At the apex of this small nenandrium, certain cells develop antheridia, and each antheridium divides transversely or vertically into two sperms. These sperms are liberated from these antheridia. The androspores are produced from the androsporangia and are just like zoospores, having a crown of flagella at the base of their beak. These androspores are produced from the main filament by the repeated transverse division of the oedogonium filament. After their release from the filament, they move freely in the water and remain in search of the female filament. On the attachment with the female filament, they germinate in attachment with the oogonium or any of the supporting cell. The germling attaches itself with the oogonium or supporting cell by means of a attaching cell, or we can say it is a holdfast. The remaining portion of this androspore germling divides transversely into a small fragment, into a small filament called nenandrium. At the apical end of this nenandrium, certain cells divide transversely and form antheridia. And each antheridium produces two sperms. The sperms on maturity are liberated from these antheridia, and they swim freely in water, remains in search of the oogonium. Now we will discuss about the fertilization of oedogonium. In oedogonium, the male and female gametes are produced by antheridia and oogonia. It is said by recent investigation that these oogonia or oogonium produce certain chemical substances which attracts the sperms. The fascinated sperms remains in search of the egg, which finally reaches the egg and penetrates into the egg cell. After penetration, probably the one sperm which arrives first makes its entry successful into the egg cell, whereas the remaining cannot enter into the egg cell because the nature of the membrane of this egg cell changes. The male and female nuclei within the egg cell fuses to form diploid zygote. This diploid zygote forms a thick wall around it and is finally liberated by the decay of the oogonial wall. Now, the germination of oospore. Prior to the germination, the oospore nucleus or zygote nucleus undergoes meiosis, resulting in the formation of 4 meiospores or meiospores, which are present within the oospore or zygote wall. These 4 nuclei are organized into meiospores or meiospores, which resemble with the zoospores having a crown of flagella at their anterior end. On liberation from the oospore or zygote wall, they develop into the new thalli of oogonium. Dear students, now we have completed this lecture. I think all of you might have enjoyed this lecture, and this might have been interested for all of you, and you may have benefited with this lecture.